This is really about manufacture of biological and biosimilar medicines. My name is Katerere and I'm at Tshwane University of Technology. So maybe a little bit about biotherapeutics and biomedicines. Let's start here. So everybody's talking about COVID at the moment. And it turns out that tocilizumab, otherwise known as Actemra, has now been approved as a treatment for COVID. The price of this product is at least $500 in around here, and I got a price in Germany which was shocking. So in Germany, it's almost 3 times the price you get it around here. And this is biological medicine. So this gives you an idea of how important these medicines are, but also how expensive they are. And in fact, this particular product is in short supply. So there's been a run on it around the world. In terms of biological medicines, so I come from a pharmaceutical era where the medicines were all small molecules. But as you can see here, we're increasingly moving towards biological medicines. So when you look at new antivirals, you look at immunoglobulins, and of course vaccines, all of these are therapeutic biological medicines. Oncology increasingly is more and more turning to highly effective biological medicines. These are just two. Then you have today. So today until the end of the day, and then tomorrow as well. And this is really about manufacture of biological and biosimilar medicines. My name is Katerere and I'm at Tshwane University of Technology. So maybe a little bit about biotherapeutics and biomedicines. Let's start here. So everybody's talking about COVID at the moment. And it turns out that tocilizumab, otherwise known as Actemra, has now been approved as a treatment for COVID. The price of this product is at least $500 in around here, and I got a price in Germany which was shocking. So in Germany, it's almost 3 times the price you get it around here. And this is biological medicine. So this gives you an idea of how important these medicines are, but also how expensive they are. And in fact, this particular product is in short supply. So there's been a run on it around the world. In terms of biological medicines, so I come from a pharmaceutical era where the medicines were all small molecules. But as you can see here, we're increasingly moving towards biological medicines. So when you look at new antivirals, you look at immunoglobulins, and of course vaccines. All of these are therapeutic biological medicines. Oncology increasingly is more and more turning to highly effective biological medicines. These are just two of the names. Then you have today. So today until the end of the day, then tomorrow as well. And this is really about manufacture of biological and biosimilar medicines. My name is Katerere and I'm at Tshwane University of Technology. So maybe a little bit about biotherapeutics and biomedicines.
We are very pleased to have so many of you joining the session today. I think online we have just over 20 participants that have joined in. Welcome, and please take advantage of this opportunity. It does not come every day, but it's ICGEB has kindly made themselves available to offer this training. As the Department of Science and Technology, Science and Innovation in South Africa, we feel it's important that we continue to support human capital development in the continent. We also feel it's important that we make accessible opportunities for training, but also the IP that can be exploited that has been developed through our joint investments as members of the ICGEB. And this afternoon we'll hear from Dr. Natasha Skoko, and tomorrow from Dr. Aguinancia and the other ICGEB colleagues. But this afternoon, in particular, the training that will be offered is on technologies for the production of biologics and biosimilars. I think a lot of people ask themselves, why would the Department of Science and Innovation in South Africa not only focus on its national interest? Why are we so heavily invested in a regional and continental value chain? We are not an island, and it's very important that as we share the health burden, we also jointly contribute towards contributing to solutions that can address the health burden that the entire continent faces. The TISA STI strategy for Africa 2024 is a pillar also that strongly supports human capital development. So does the SADC industrialization strategy, which also has a pillar that speaks to pharmaceuticals value chain. Our own national strategic documents, such as the 2019 white paper, the current draft decadal plan, our national development plan, all speak to a capable human manpower for us to not only address issues related to health, but to also address the sustainable development goals. So for us, it's indeed an honor and a pleasure to have the ICGEB co-host this event. And I would not like to waste any more time because we have lost some bit of time, but to immediately hand over to our facilitator, Dr. David Katerere. Over to you, sir. Thank you.
Thank you very much, Mampe, for that kind introduction. So we'll go directly now to Dr. Spoko to give us the first presentation. Over to you, Natasha.
Okay, thank you very much. Thank you, David, and thank you, Mampe. I'm really happy to be here. I'm sorry for this delay, but I will try to be fast as possible to present and send you a message that we prepared today for all of you. So I will now share my screen. Okay, if everything is fine, you should be able to see it. Okay, so as you heard, I'm heading the Biotechnology Development Unit at the ICGEB in Trieste, and today I will speak about biologics, biosimilars, different expression system, production process, our biologics portfolio and technology transfer model, and the product process development. So just a quick overview, the ICGEB is International Organization for Research, Training, and Technology Transfer to Industry, Life Sciences, to promote sustainable global development. We have 3 centers in Italy, Trieste, in New Delhi, India, in Cape Town, South Africa, and the headquarters is here in Trieste, in Italy. So we operate within the United Nations systems, and we have 66 member states all around the world. When we talk about ICGEB instruments of action, we are basically talking about different scientific research in our laboratories in 3D centers. We are talking about the training, fellowships, organizing different meetings and courses. But last but not least is a transfer of know-how to industry for the production of biologics and diagnostics. So this is exactly where my group works in this field. So biologics or biopharmaceuticals. We heard from David that these are very important medicine produced from living organisms using biotechnology techniques. According to European Medicine Agency, biopharmaceuticals may be recombinant proteins. Just to give you an idea what we are talking about. We are talking about recombinant protein, monoclonal antibodies, different vectors, antibody fragments, nucleic acids, oligonucleotides, and vaccines. Today, more than ever, we all talk about vaccine and the production of these very important biologics. Biologics have been introduced in 80s, and now there are more than 350 biologics on the market, and thousands are under development. Like you can see here in this chart, over and over again, we have more and more identified targets, new hits, and new molecules coming out. Biologics, we would all agree that biologics have revolutionized the treatment of anemia, diabetes, autoimmune disease, and cancer in the last decades. Here just a brief overview telling you that, of course, these biologics are one of the fastest growing segment of the pharmaceutical industry. And here you can see what does it mean in the numbers. So this year it was predicted, and I think we hit that the global sales revenue, it's like around for all biologic medicine is around 350 billion euros. So monoclonal antibodies are the largest segment in the terms of revenue among all the categories of biologic drugs. Biologics. So just to have the brief reminder what we are talking about. We are talking about the blood factors, hormones like insulin, growth hormone. We are talking about erythropoietin, hematopoietin growth factors like erythropoietin, GCSF. We are talking about interferons, monoclonal antibodies that are used now in the treatment of cancer, autoimmune disease, Alzheimer. You probably heard lately that the FDA, after so many years, finally approved the first biological medicine, adacanumab, monoclonal antibodies against the Alzheimer, a few months ago. And then also David mentioned tocilizumab, like monoclonal antibodies against COVID-19, but there are plenty, at least 4 other monoclonal antibodies that are now in clinical trials against COVID-19, against the SARS-CoV-2. And we are talking also about the vaccines. So biologics have tremendous health benefit, but there is an enormous need for them. But most majority, they're unaffordable to the majority of people on the earth. According to as you can see here, unfortunately, like just if you talk about the old disease like diabetes, according to the World Health Organization, people with diabetes will more than double over the next 20 years, and they will reach total of 366 million by 2030. And it's very important to say that most of this increase will occur as a result of the 150% rise in developing countries. So as you can see here, in this study published by medical journal Lancet, has predicted that sub-Saharan Africa could see an increase of 85% in the number of cancer cases in 2020. So yes, there is a huge need for biologics. When the biologics patent expire, then they can be produced as a generic of biologic drugs, but we don't call them generics, we call them biosimilars. So this is a very nice classification. I just wanted to show you, just to remind you how the original and the copy drugs are divided. So you have an original true innovator, you can have a biobetter, like it's a Pegasus pegylated interferon alpha, and you will hear something more about this molecule and other modified molecules by my colleague, Dr. Guarnaccia, later. And then we are talking of the non-originals, we are talking about biosimilars. So let me show you biosimilars. So biosimilar is biological medicine product that contains a version of an active substance of the already or authorized original biological medical product, reference medical product, but it's very similar. It's a new version of existing biologic following of loss of the patent production. Why similar and not equal? Because every small, this is a overall overview of the production process of one biological drug, and it includes the upstream processing, like the preparation of the cell culture, production of the molecule, then the downstream processing that includes purification, then you have a formulation and all the quality control tests. Why similar and not the same? Because every small change or difference in manufacturing process in all these paths may have a significant impact on the quality, purity, biological characteristics, activity, of the final product. So we have to search to have as similar as possible product to the reference medical product. Similarity to the reference is in terms of quality characteristics, biological activity, safety, and what is really important, efficacy. And everything is based on the comprehensive comparability exercise between molecule biosimilar and the reference product. Biosimilars are developed, manufactured according to the same quality standards as the original product, and manufacturing process, including cell lines, production strains, they have to be developed to guarantee comparability with the original product. So the main drivers of biosimilar growth, they are, first of all, we said a patent expiry of the original biologics. Then the second is budget constraint on the healthcare system. We mentioned that the majority of the people cannot afford them because they're very expensive. So it's a huge constraint on the healthcare system in the countries, and limited patient access to expensive originator biologics, particularly in developing countries. So biosimilars are around since 2006. The first generation of biosimilars came with the first expiring of the patents, and they include interferons, GCSF, erythropoietins, growth hormone, insulin. Second generation of biosimilars are here, of course, since 2013, and they include the monoclonal antibodies, protein generated by fusion of antibody receptor moieties, and so on. So here you can see how the patent for all these monoclonal antibodies or other biologics expired. So then we have them now available on biosimilars. So it's the loss of exclusivity that drives biosimilar interest. So all these products already lost the patent by last year, and here you can see all these monoclonal antibodies that, like the adalimumab, rituximab, trastuzumab, they are now available as biosimilars or are in the development stage. So what's next? After the second wave, we will have the second generation, we will have a next wave. In the current wave, biosimilars have been limited to the inflammatory diseases, oncology, diabetes. But in the next wave, if you see here what are the molecules in the development, we'll see biosimilars enter different therapeutic areas, neurology, respiratory disease, ophthalmology, and we also mentioned Alzheimer's disease, we mentioned now COVID, and so on. So impact of biosimilars, since they were introduced in the European market in EU 5 countries, their price drastically dropped. So the drop in the price can be up to 50 to 70% due to introduction of biosimilars. So they're cheaper, and as they're cheaper, they also allow to increase the volume of the treatments per day, so many more patients can be treated. So this is experienced in European Union, but this is more or less experienced everywhere. In UK, entry of GCSF biosimilar to Neopogen has increased patient success by 50%. So it means, and you can see here how many patients can be treated more, and better economic perspective translates into better health perspective. Our guests from Mexico, the people that we have in Mexico, they also provide us this graph where we can see that due to the erythropoietin biosimilar introduction in the Mexican market, average cost price of the erythropoietin drastically dropped, and that drastically increased the access of erythropoietin in Mexico. The same, more or less the same situation was with the rituximab. So we know that now the price can drop by introduction of biosimilars, but there are different legislation and guidelines to follow when it comes to the registration and development and registration of biosimilars. So you can see here all the biosimilar landscape and how FDA, EMA, and European Medicine Agency and World Health Organization work on these biosimilar guidelines. And they're constantly working to improve, to put the new guidelines and to help countries to register new biosimilars. So guidelines for biosimilars, they basically, as we said, they include 3 different levels. First level has to deal with the quality issues. So guidelines on similar biological medical products contain talk about the quality issues that address all the comprehensive side-by-side quality analysis that you have to perform in order to show that that biological biosimilar is as similar as possible to the reference standard. So these are all analytical considerations that you have to have when you are showing the quality of the biosimilar product. So we have to see the primary and higher order structure, amino acid composition, post-translational modification, biological activity, purities, and so on. So a comparison of the biosimilar to the standard following just pharmacopoeia monographs that are first monographs to follow this quality, it's not sufficient for the purpose of comparability. The biosimilar, but it's the first step. The biosimilar should be demonstrated to be similar to reference medicine product approved in the country, which is selected by the companies developing the biosimilar. And after doing all these different tests, of quality tests, so there are more and more analytical techniques and tests necessary to show in order to go to the second level, that is non-clinical trials and then clinical trials before registration of the biosimilar. So just a simple classical demonstration is the erythropoietin, the molecule that has different glycosylated forms, but the different producers, what you can find in the market, produce different quality of erythropoietin. So even slight changes in the growth of the cells, purification, and so on, can drastically change the quality of these isoforms, the quantity and quality of these isoforms, that will impact the biological activity of the erythropoietin. So if we followed all these guidelines, we will see that if the first part is done, all the quality test and comparison with the reference standard, then the companies can go further to show in a non-clinical studies and the clinical studies that the drug is similar, can be called biosimilar, and all of them, they become the part of the totality of evidence that the medicine agency in each country should look together. So FDA scientists, how it works. So FDA scientists, for example, or EMA or SAFRA or whichever agency, the local agency is responsible for that, they will evaluate the applicant's integration of various types of information to provide advance on a scope and extent of development plan, and an overall assessment that the biological product is or is not biosimilar to the approved reference product. So all of these they have to put together and then assess the biosimilar. So Sandoz somatropin growth hormone biosimilar was the first one authorized in European Union 2006. An additional 55 applications have been approved in Europe until today. And filgrastim biosimilar received the first USA approval in 2015. So we can see how United States were much later came with the biosimilars regulation and registration on their market, much later than European Union. But until today, FDA has granted 26 approval for biosimilars, with 11 being currently on the US market. Apparently, what I can find, it's in the literature that Teva pharmaceutical industries has become the first company to launch the biosimilar drug of filgrastim in South Africa in 2008, marketed and distributed by Cipla. So more and more biosimilars are coming out. So I would like just to spend a few minutes to tell you about the manufacturing process. So as we said, we start with the genetically modified genetic engineering. We insert the gene to produce some biological drug, like a gene for human growth hormone. Then we do all the cloning and preparation of the cell bank. We prepare the master cell bank, and then we are going to the gene expression in the bioreactors. After that, we purify the protein to the different level and the different chromatography steps to have a good formulation of the product, and also do all the quality control tests. So a large-scale cultivation of genetically engineered host cells, which harbor artificially transfected gene for the protein of interest, is the first part. So it would be nice to have a single expression system with an optimum blend of all protein expression features and parameters, but however, such system it does not exist today. So we have different systems, and for different molecules, they are suitable or not. So the main expression systems that we use today in biologics to produce biologic drugs or biosimilars are bacterial system, E. coli, yeast, Saccharomyces, Pichia, Hansenula, or mammalian cells like Chinese hamster ovarian cells. So what here we can see, okay, and now more and more monoclonal antibodies are coming to the market. Mammalian expression system is starting to be even more used than the microbial that was traditionally the favorite system in the biotech companies. So even when we talk about vaccines, COVID-19, whatever you hear here about the production by bioreactor, or we can mention vaccines for COVID-19. So you all heard about 3 different technologies to produce different vaccines today that are registered and already on the market. So it's or inactivated vaccines, so that contain inactivated virus, like Sinovac vaccine, or a viral vector adenovirus vaccine that is designed to create immunity using adenovirus which get genetically modified with insertion of coronavirus protein. Or the third newest technology is the messenger RNA vaccines, that is artificially produced mRNA in the laboratory with the aim to train our immune system against the virus. So why I'm mentioning this? Because all what we learned from the biosimilars, biologics, it can be applied. Of course, it was applied here in sense that if you want to produce artificially mRNA, you have to transcribe it from the DNA, and it's a plasmid DNA that you have to prepare it in the bioreactors in E. coli. So you transform bacteria, and then the cells, you grow it in the large scale in the bioreactor. So, and then from that you obtain the high quality and good quality, the high purity of the plasmid DNA that then you use to produce the messenger RNA. So all these things like start in the upstream process, as I told you. So to produce the different cells, we can use the bioreactors with different size. Of course, when we start in the lab, we start with 1, 2, 3 liters, then go to 30 liter, and then further 300 and even 3,000 liters. So challenges today in the biologics and biosimilar development, vaccine development, it's really like how to produce a lot, like produce the milligram scale. So moving from microgram to milligram doses of different recombinant protein therapeutics really has changed the face of biotechnology. Because if you look in the 80s, like what was in the 80s standard, then to produce like 100 milligram per liter of bioreactor broth, today is not anymore, it's not good enough. Today we can go up to gram per liter scale, and this is where all the challenges come. So this is how we have to be very fast, very efficient to have a highly producing cell lines in order to produce a lot of plasmid for mRNA vaccines, to produce a lot of cells for other biologics. So cell line development, traditionally it took a lot of like months to produce, from the gene design, for example, if you're talking about mammalian cell, to the cell banking. But today, luckily, with the new techniques, with the robots that are helping us to do a fast clone picking, to grow the cells in the parallel bioreactors, this time for 6 months can go down, can be done much faster. So after the production and upstream processing, we are going to the purification part. Purification part includes different chromatographies to purify the protein. And what we do in the lab during the development, of course, we all start with these tiny columns and then we go through scaling up to columns that are industrial columns with a few liters of resin, where we can purify gram scale of protein, and going up to 30,000, 300,000 doses per batch. At the ICGEB, we have developed this model for, we have developed first all of this expertise to work and develop the technology to produce biologics, and then we came out with our model for technology transfer. So why we would like to do that and why it's important, and this is really like somehow the take-home message, because we really want to strengthen capacity and competence of the local producers to accelerate the product availability at more affordable price in the local market. We want to help to decrease reliance of imports and help to treat conditions with high incidence in the population, to have more trained bioprocessing professionals in the emerging market. Why it's important? So what COVID pandemic teach us? If you look in the African landscape, more than 90% of biopharmaceuticals, for example, in South Africa is important, including vaccines, MAPS, insulin. More or less the situation is the same, if not worse, in other African countries. High prices of off-patent biopharmaceutical medicines, simply it's because these products do not have competitors from biosimilars. Shortage of trained specialists. So these are all the things that has to give us the message that the local production is essential. That the South African and that the like many of these suppliers from outside, they really stopped supplies during the COVID pandemic. So what we learned very fast is something that here we at ICGEB and Biotechnology Development are insisting for last 25 years. Like, yes, let's have a professionals, let's have a knowledge in-house so we do not depend on the imports. Biosimilars in South Africa. In South African biosimilars guidelines is essentially based corresponding to the guidelines of European Medicine Agency, World Health Organization, and they're now more or less all harmonized. In some cases, collaboration with local producers are really critical for success. And for example, as I mentioned, in South Africa, Teva has established strategic agreement with Cipla, a South African manufacturer, to launch the country's first biosimilars for the oncology hematology market. What I can find in the literature, but I would like to confirm it, that the two biosimilars have been approved in South Africa, Filgrastim and Trastuzumab. So how we at ICGEB help this and how we help to move the biosimilar market in our member states. So we have developed in-house expertise for production of biologics. We transfer this know-how with no exclusivity. We really have this training in-house and knowledge sharing. We developed all the quality control procedures in accordance with European medicine, European pharmacopoeia monographs, and we offer it in an hour, we call it technology in a briefcase. So we really, if you remember at the beginning of my talk, I talk about a lot of analytics, a lot of the tests that has to be done when you develop your biologics. So this is exactly comparable analysis, quality control test, product development that takes few years. Every single company needs to spend 2 years before going to the process, scale up development, non-clinical and clinical trials. So what our idea was that when we develop one technology, we want to save companies few years time on this first stage, so they can easily and faster moving in the second stage. So in biotechnology development unit at the ICGEB, we have an expertise in upstream, downstream processing. We work with bacteria, yeast, and mammalian cell. We have fermentation facility up to 30 liter per hectare for microbes, or 2 liter for mammalian cell. We have expertise in all kind of chromatography for the pilot scale purification. We did this for our industrial and our partners. So from the lab scale, we scale up some of the technologies to the pilot and pre-industrial scale. So scaling up for them, then it's much easier to the industrial scale. We have developed the synthetic organic chemistry laboratory where we do chemically modified biologics. And this is what Dr. Aguinancia after me will talk about that. We will talk about polymer chemistry, peptide and protein chemistry, and protein conjugation with these polymers. Then we perform all these quality control tests. We follow the European Pharmacopoeia guidelines for specific for each product, and we can offer training in all these technologies. So over the 25 years, we are really happy to say that we develop all these technologies in-house and offer them as a technology transfer packages to many countries. So GCSF, interferons, practically all the first generation of biosimilars. And for example, I will give you just one example, that this is the whole production process, starting with genetically modified cells, as I told you, bioreactor of 30 liters for yeast. We are using Pichia pastoris, and then purification and finishing with a concentrated protein solution. So for example, insulin, why I mentioned at the beginning so much, because there is not just COVID pandemic, then there is diabetes pandemic. So it's predicted that the current doses of insulin are not enough to cover all this need. So we developed the technology to produce insulin precursor and in a yeast Pichia pastoris, and from that one molecule we can finish with 3 different molecules. At the end of the process we can obtain short-lasting insulin or 2 long-lasting insulins, Katerere and Biologics. So this is basically just to show you that you have to do the fermentation, follow every day the peak of the insulin precursor in different stages of purification to arrive to the to purified protein. So just to give you a brief idea, what does it mean to develop it? So we have more than 25 years experience in developing this. So it's really develop and have to put also innovation in the purification part, which kind of column, resin you will use, and always keep in mind to have it economically viable process. So we work with more than 25 years with the countries, with our member states, and always try to have the best quality, but also the best price of all this material used in the production process in order to decrease the price of the drug on the market. So this is what I told you, from insulin precursor, we finish with the insulin or 2 chemically modified insulins, Katerere and Biologics, and Dr. Aguinancia will tell you more about these interesting molecules. So steps in technology transfer and training, it's very simple. We finalize the technology transfer agreement. It's very straightforward, and then we are free to share knowledge. We usually, before COVID, we would receive people from the companies, universities, startups, whoever wants to teach. So really, it's no exclusive knowledge transfer, to stay with us up to 6 weeks in our own laboratories, and then they can have a hands-on experience in the production, purification, quality control test, and then we give them all the protocols and all the material generated here. Because of the COVID and pandemic, we really had to react very fast, and then some of these technologies for the production of insulins, we generated video-based training, and now we offer it as online technical assistance and video-based training. Here you can find my contact details and our newly launched web page. We, as you heard, we had more than 70 agreements with companies from 22 countries. We trained more than 100 scientists trained in our own lab. And here in this world map, you see in blue are the countries where technology has been transferred, and in green, countries where technology has reached market. I'm really happy, and this is something new. Since the beginning of the COVID, we really work hard to build a new facility. So with a grant of 3 million euros, grant from the region Friuli Venezia Giulia, where Trieste is based, we decided to invest and to build a new pharma-compliant laboratory at the ICGEB that are clean rooms in the class D and C that could be, in order to be able to develop the technologies for the production of the second generation of biosimilars, monoclonal antibodies. So our new laboratory is comprised of the mammalian cell line development facility, upstream, downstream process laboratories for microbes and mammalian cell, chemical lab, polymer preparation lab, and the quality control labs for biologics. So this is how our new facility looks like. Why we wanted to build this, to be able to have a better clean environment to develop the master cell bank to produce monoclonal antibodies, and also to have a new equipment that we can do a new quality control test that are requested today for biologics, and what is more important, to train people. So what we learned here, and you will hear it tomorrow, I would really like to invite you tomorrow to listen to Dr. Terdoslavic presentation about good manufacturing practice and quality assurance principles that we've been trained over the year, and she can tell you our own experience. So how these very dry documents of the GMP good manufacturing practice documents can be really applied and put in practice. And this is what we want to offer as a training as well. We want to share our own experience, how it's possible to build some facility like this, and how this facility can be copied anywhere in our member states, and even in the small entities, the small startups, universities, or governmental institutions, could be the idea of the transfer this kind of knowledge to prepare their GMP-like laboratories and to train professionals and have something like this in their own countries. So this is how our fully equipped, in the 11 months, we constructed all these laboratories over the 215 square meters, and we also bought a new equipment that will help us to speed up the development process. So I would like to take the last 3 minutes of your attention and to show you our video, how this laboratory looked before, how the building looked before we took it before the March last year, and how it looks now when we are already in and fully active. So I will show you this video, and I hope you will enjoy. last, I would like to show you my team. So we are a very small team, but very efficient, and the people are very dedicated to this mission of the ICGEB, that is the development and the knowledge sharing. So here you can find our newly launched website, where you can have all the information about the technologies, about our work and our mode of action. And this is my email address, so you can contact me directly. And thank you very much for your attention.
Thank you very much, Dr. Skoko. Well, I just want to find out if there are any questions, and people can actually be typing questions and comments in the chat box. I just wanted to ask, so in the US, you said there have been 26 biosimilars approved and only 11 are being marketed. Why is it that the minority or a smaller number is being marketed? Is it a market dynamic issue?
I think it is, and also we have to think that we have to keep in mind that also in United States they started like 9 years later than European Union. So in European Union we accumulated almost a decade of the all the facilities, you know, the production, the regulation, and everything before United States. So yeah, I was also surprised, because I was expecting that as soon as you got it, you are going to produce it, but it seems they have some issues about that. But anyhow, the application number of applicants, it's also a lower in comparison to European Union, but it's fast. It's anyhow a very fast and dynamic issue.
Okay, and then just one thing that strikes me. So in Africa, we produce a lot of life scientists, you know, biology and biological sciences, biochemistry. In the university, they are very popular. But so from capacity building, HR development, training, I think we are fine. But the biggest barrier I see from your presentation is the equipment and infrastructure. So what creative ways would you suggest for industry to use to be able to get into this space and still have some money, you know, left over?
Yes, exactly.
Yeah.
So you are right. I have no doubt that the students are generated, but what I can see also here in Italy, we have many generated students, but not a practical bioprocessing training. So this is, you're right, you need to have a facility where you would train people. So what we see here even, that the student come to us and they said, yeah, can I just enter in your facility just for 6 months and be trained, just to have an idea after the good university theory and everything, that good training at university, but not in the bioprocessing. So there is a really huge gap between the university reality and the working reality. And for that we saw, and that was one of, you're right, that was one of the reasons why we asked the region to build this facility, because we said the region, the government, anything, it has to recognize that there is a need for even not private, because we are not private, not profit organization, where the young bioprocess engineers, the people can be real trained and prepare for a real world. And I can tell you our experience, like 2 years ago, we trained people from the company here, biotech company in our region, and they really struggled to build a team. So we said, great, give us 4 people, you know, to be trained. And they had to announce and search for more than 6 months the people who has experience in it. And then we saw, and we started to talk about this triangle and said, look, can we be a training center? Students from the university pass through our center, and then they are ready for the industry and for real work in the bioprocessing field, in the biopharma. So
Okay.
I think it's
Like
really critical.
Yeah.
This is critical. And of course, as a private sector, they want to have the people ready to start to work, produce.
They're very impatient. Yeah. Okay. Yeah. But thank you very much, and we look forward to sort of further engagements and presentations later on, and also tomorrow. And I think the audience has your contact, so let's take this opportunity on board and see how we can build the African biosector really. Okay. Thanks a lot, Natasha. So next, we are going to have Dr. Corrado Aguinancia.
Yeah. Hi, all.
Corrado,
Hi.
over to you. Good to see you again.
Thanks.
And
Thanks.
thank you.
Okay, I'm ready. I will start sharing my screen. Just a second. Okay. Okay, I hope it's visible to everybody in a few seconds.
Yes, we can see you.
Yeah, you can see that. I don't see that. Okay, now I started the presentation. Okay. So, welcome to everybody. I will tell you something about the experience of my lab, the Biotechnology Development Unit in ICGEB, about a branch of what are the biologics and biosimilars, so the molecules which are not only produced by recombinant technology, but then they are also chemically modified to change or improve some of their characteristics. Okay, I will just recall few informations also from what already Natasha said about biosimilars and biologics. And first of all, comparing the complexity of the molecules of different therapeutic classes. So here you can see some molecules which are practically generic, so small molecules like aspirin, paracetamol, antibiotics, and so on. Then you can see the already increasing complexity in some molecules which are on the border between the organic world and the biological world. So molecules like, for instance, insulins, or in this case, one of the peptides which is composing liraglutide. And then the increasing complexity of biological molecules like such as erythropoietin, or eventually also monoclonal antibodies and so on. And so biosimilar, as Natasha says, is a biological medicinal product, which is the new version of a biological which is being already authorized by regulatory agencies. Okay. The important of biosimilarity is the fact that biological products should be highly similar, with the minor differences only in clinical inactive components. Okay. And there are no clinically meaningful differences. So from the point of view of the biological activity in the organism, there shouldn't be any evident difference with respect to origin. I will introduce a concept. The concept is the one of the so-called critical quality attribute, which is very important. So is the characteristics from the point of view of the chemistry, really, of the molecule, of the biologics, which can affect the quality of the biological product. Okay, so this critical quality attribute must be evidenced from the very beginning when you are projecting the creation of a biologic or a biosimilar, because they will be critical during all the process, from the upstream to the downstream to the final packaging of the drug. Okay, because these chemical characteristics can affect the pharmacogenetics, or the efficacy of the molecule, these modifications like the amidation, oxidation of amino acid, in terms of sequence, glycosylation of the molecule. So there are many of these characteristics which are important for the point of view of the safety and efficacy of the drug, or the final biological drug. Here, for instance, you can see which are the possible sites in the monoclonal antibody which are important, for instance, for the biological activity of a monoclonal antibody. So you can have heterogeneous terminals with a chemical modification which is called glutamate. You can have exchange of the disulfide bonds which are keeping together the molecule. You can have the amidation, oxidation, which are affecting methionine and asparagine residues. You can have cleavage, so you have fragmentation of the antibody, which is not more active as the original molecule. You can have differences in glycosylation and so on. So many many different characteristics must be controlled during the process and in the final product after production. What is also very important that when you start a project for creating a biosimilar, the information about the reference product, so the originator molecule, is not publicly available, okay, because it's proprietary and is being given only to the regulatory agencies, okay? So you have to start practically from scratch. You have information for starting the process for creation of biosimilar. So you must develop what can be also an entirely new customized project, okay, process, okay? So that's why you have to very clear which are the quality attributes that you want to monitor, which are the parameters that you want to monitor during the process, okay? There is a this is a summary of what is a typical process for creation of a biologic or eventually also biosimilar, cell line creation, cultivation of the cell line production, isolation of the product from the media, usually, or from the inside of the cells which have produced the molecule, and then you have formulation, fill and finish of the medicinal product. Okay, and I give you just two examples of what is a very important part, which is the purification of the molecules that you are going to produce. Here are some examples. You know that the bacteria, the factory, so either the bacterial factory or the yeast or the mammalian factory have differences in the way they produce the biological molecules. And so you have differences also from the point of view of the downstream processing. So how you purify the biologics that you are going to extract from them. So you can have, for instance, for products expressed in E. coli, like the GCSF, of which we will speak a little bit later. You can have, for instance, you have to disrupt the E. coli cells, then you have to completely extract by denaturation the GCSF. You have to clarify it and subject it to a process which is called refolding. So you have to rejoin the disulfide bonds, and then you have to subject it to a various type of chromatography. Instead, in the case, for instance, of products like insulins or growth hormone, which are produced in yeast platforms like Saccharomyces cerevisiae or Pica pastoris, you can have the product, the biologic, which is already present in the media, in the refolded form, so it's already in the final form from the point of view of its primary, secondary, tertiary structure. So you have simply to extract it from the media and then subject it to several steps of chromatography to clean it to achieve a pharmaceutical purity. Maybe you've seen already this type of picture from in nutshell presentation. So it's just to show you the difference between which is the pathway, the regulatory pathway, which biologic have to be subjected. In the case it's an originator, so a new biologic product, or it is eventually biosimilar to something which is already planned for market. As you can see, while the clinical phases are much more present in the case of an originator, in the case of a new biological product, for which you don't know nothing about especially safety and efficacy, you have much more burden from the point of view of analysis in the case of development of biosimilars. So the highest number of analytical tests serves to demonstrate the similarity from the physicochemical point of view to the originator, and so to establish already a good base to affirm that the final molecule will be highly similar to the originator biologic. Okay, this is, I don't pretend that you're going to read, but there are, for instance, from the point of view of the critical quality attributes that you have to monitor, there are 3 pages like that of analysis in various type of instrumentation, various type of chemical physical parameters that you have to assess before saying that a molecule is really similar to an originator molecule. Okay. From its point of view, anyway, European Pharmacopoeia is coming in help. There are monographs for many biotherapeutic products. There is obviously also a US Pharmacopoeia, or there is English UK Pharmacopoeia, there is Japanese Pharmacopoeia. So generally the Pharmacopoeia are helping because they are public standards. They give you monographs and by which you can assess a certain number of critical quality attributes attributes. And if you are not, if your product doesn't pass some of these tests of these monographs, you can already say that probably your biosimilar is not good and can be excluded from ever reaching the market from this point of view. Okay. Anyway, simple compliance with the monograph doesn't mean that you are fully biosimilar to the originator. Also, I want to say that in GMP regulatory framework, all these analytical tests must be done in a special approach, I mean, with all the instrumentation which has to be subjected to a series of tests like IQ or Q and PQ, so installation qualification, operation qualification, performance qualification. Okay, so also the instrumentation that you're going to use must be certified that is able to perform all the analytical test in the proper way. And this is actually what we are trying to do now with our new facility. Okay, this is just an example of the number of tests which have been performed erythropoietin just to assess all the parameters which are necessary for the quality of the molecule. Okay, I will come quickly to the real subject of this talk, which this is a list of the products that are actually technologies that are actually developed in our group in ICGEB Biotechnology Unit. You can see interferons, granulocyclone stimulating factor, erythropoietin, and insulin as anchor hormone. In particular, we'll talk about some of them which are modified. You see like PEG interferon alpha, GCSF regulated, PEG-EPO, and this couple of insulin. I will just take a couple of examples. I will not speak on all of them for reason of time. So why we should chemically modify a biologic or a biosimilar? So most biopharmaceuticals, as you know, are based on endogenous proteins which are present already in the organism. So they are hormones, enzymes, antibodies, growth factors, interferons. Sometimes when you inject in the body some of these molecules, they are not able to perform their biological activity in the proper way because by parenteral administration, they undergo a quickly removal from the organ. Okay, this can be done by renal ultrafiltration, especially in the case of small proteins. If they are small proteins can be too small to pass, they pass through the glomerular filtration of the kidneys. They can be digested by proteolysis by peptidases. They can be lost by insolubility absorption. They can be eliminated by immunogenic system. So the chemical modification is a way to improve the half-life, the stability, and sometimes also reduce immunogenicity or increase the solubility of these molecules. Okay, I will talk about two of the main strategies to improve the pharmacogenetics, pharmacodynamics of the biologics, which are the polymer conjugation, in particular the pegylation, and the lipidation. Okay, so conjugation with polymers of protein glycol and conjugation with fatty acids. Okay, then there are several other methods, like you will see, I will show you a small example in insulin, protein sequence modification. You can improve the glycosylation to make the molecule bigger. You can fuse it with larger fragments, like, for instance, albumin or pieces of monoclonal antibodies and so on. So there are several strategies, but I will talk to you about the first two. This is a table which shows you the current approved peptide and protein biopharmaceuticals, which are pegylated or lipidated. Okay, all the green molecules, these are pegylated molecules. Like, we have developed most of them actually, because these are second generation biologics, so PEG-interferon alpha 2B, alpha 2A, PEG-filgrastim, pegylated erythropoietin. We are actually developing certolizumab pegol, which is a piece of antibody pegylated. We have, and also these are instead molecules which are lipidated. So you have a second generation of insulins and a peptide which is called liraglutide, which is also used against obesity and diabetes. Okay. So let's see exactly what is polyethylene glycol. Polyethylene glycol is a polymer. It's generally regarded as safe by FDA, so it's approved also for human consumption. It's water soluble. It's biocompatible. From this point of view, is not very immunogenic. Sometimes can be immunogenic in some cases, okay, but it's there are several strategies to decrease these type of problems. There are actually 7 approved therapeutic proteins PEG conjugates and 1 oligonucleotide PEG. So it's this is an RNA aptamer, and PEG is mainly affecting the molecule from the point of view of the fact that it's increasing the in vivo half-life by a simple mechanism of increasing the size, the hydrodynamic size of the molecule. And it can also enhance the drug performance because it makes it more soluble and makes a kind of shielding action for the against the immune system. Okay, so it can decrease sometimes the effect that the immune system can have on the molecule. They destroy that the fact that the immune system can destroy the molecule when it's injected. Pegylation can be done in several sites of the biologics. So many amino acids can be pegylated, so can be used for pegylation attachment. You can have enzymatic pegylation. You can pegylate on the thiol groups of cysteines. You can pegylate on disulfide bonds. You can pegylate on C-terminus, on the sugars. You can have non-selective amino pegylation. You can also have very selective amino pegylation, like in the case of N-terminus. Okay, so there are several chemistries which are allowing the thing. I will not enter in details, but just to show some of the chemistries that we developed in ICGEB for pegylation of amino groups, either site-selective, like in this case, or not site-selective, or in case also branched PEGs or complicated molecules where you can have 2 PEG polymers connected to a branching site and then a single point of attachment on the molecule. This is the case of one interferons. Okay. In fact, the interferon alpha-2A, for instance, is one of the molecules that we developed in-house. Or for this one, we arrived up to pilot scale, gram scale, practically. It was used until a few years ago, mainly against hepatitis C, but it has also some other indications from some types of cancer. And we developed in-house the full process from the production of the recombinant interferon alpha-2A, so by E. coli expression, recovery from inclusion bodies, refolding and purification. We developed in-house also fully the PEG reagent, which is a very particular PEG because it's branched PEG with one lysine in the middle. We arrived up to 200 grams of scale. And then the final pegylation process by which we can arrive up to 5,500 doses in a single batch. So just to tell you, with really a few batches of this process, we can cover the necessities of a country like a medium-sized country of several millions of people from this point of view in a pilot scale like this. A second molecule very important, which is even now has a very strong market. I think 2020 market for this molecule was around $2 billion actually. So more than 500,000 doses only in the United States, is pegylated granulocyte colony stimulating factor, which is called also pegfibastim, or with the originator name Neulasta by Amgen, is a molecule which is used to reduce the duration of neutropenia when people is having cytotoxic chemotherapy during cancer. The pegylation has completely changed the parental profile of this molecule, because with a single dose you can cover the full cycle of chemotherapy. Instead, before pegylation, the simple G-CSF injection should have been injected once a week or even. Okay. Also for this molecule, in ICGEB, we developed the full process with the production of the recombinant G-CSF. The full synthesis of the reagent. This is a very special reagent because it's site-selective. This is an aldehyde, which is site-selective to the N-terminus part of the G-CSF molecule. And the pegylation process scale is around, again, with a gram scale. And in 4 weeks, we can have gram scale of the final active pharmaceutical ingredient of the pegfibastim. I will enter a little bit more in the detail, just to show you that when you produce a biosimilar of this type with increased complexity of the fact that it's chemically modified, you have to have full control of all the process. So this, for instance, in-process controls for the pegylation reaction. So when you put the pegylation reagent in contact with the molecule, you can see here I pick this actually the product of interest, so the pegylated GCSF, which is produced already in the crude in high yield, more than almost 82% of the GCSF is pegylated. Then we will have to purify by preparative chromatography. There are several steps of chromatography of different types, which can be applied to obtain the final molecule. And then you have, as I told you before, a quality control, which can be very extensive. I will show you just few data, like for instance, co-injection with the originator. You can see the red line is the originator of Neulasta. This is an HPLC, high pressure chromatography. So this is the originator, and this is the blue line is our biosimilar. And the same for SDS page. You can have originator and our biosimilar in comparison to different type of staining, both for the protein part of the molecule and for the polyethylene glycol part of the molecule, just to show you that our molecule is very pure, also in comparison to the originator, which has a little bit of higher bands. And then we obviously check also, since I told you this is a site-selective pegylation, we check by mass spectrometry the PEG moiety as in effect going to the N-terminus of the molecule. And finally, we also obviously the bioassay, which is showing the biological activity of the molecule in vivo in the cells. And you can see perfect overlapping of the standard, which is given by the originator molecule by Neulasta, and our biosimilar, which is the biological one. So they are perfectly overlapping. So the stated potency is exactly the same for the two molecules. Okay, I will make a very short deviation talking about formulation for biologics, only because this is important to introduce some part regarding insulin, so which I will talk in the final part of my talk. So obviously stability of biological molecules like peptide proteins, monoclonal antibodies, vaccines is poor in comparison to what we know as small molecules like aspirins or any other antibiotic or whatever, since the molecules are much more complex. So you have a full list of excipients which should be added to the final formulation, either to stabilize or to sometimes also to change the characteristic of the products. So you can have different buffers, different amino acids or stabilizers like lactose, trehalose, sorbitol, and so on. You can have surfactants to diminish the stress that the molecule has when it's in contact with the atmosphere or in the interface with water atmosphere. You can have antimicrobial to avoid the growth of microorganisms inside. You can have metal ion circulators, like in the case of insulin, we will see the role of the zinc ion. And so many other of these excipients must be tested, and they must be tested in wide ranges of formulation. Most of the time, when you are starting with developing a formulation of the biologic, you have to make a kind of design of experiment for checking which are the parameters in which range actually they are affecting the stability of the molecule. So type and concentration of the buffer, pH, different type and different concentration of stabilizers, salts, and so on. Okay, so it's really a science apart from this point of view. It's even more complicated when we're talking about vaccines, because vaccines can have antigens of different types, like live viruses, inactivated, purified recombinant proteins, purified viral proteins, toxins, polysaccharides, and so on. So, and then the fact that vaccines must be administered to large population in many different conditions makes the fact that the storage requirements, so especially the maintaining of the cold chain, is the main trouble in this case. Okay, so formulation process very often for vaccine is based on the preparation of solid dried powders by spray drying or freeze drying, which should be eventually reconstituted, like is happening now, for instance, for COVID vaccines. And so you need to select excipients which are protecting actually the molecule in the dried state, and so you have to choose some of the excipients which are most used, for instance, this lactose, trehalose, mannitose, and so on, which are employed in formulation development. Vaccine obviously doesn't have to contain only excipients, but it has to contain also adjuvants. Adjuvants are additional components for enhancement of the immune response. So you can have very common aluminum-based adjuvants, which are actually the most common class of adjuvants, or you can have also many other types, very often aldehyde phospholipids, saponins, viruses, and they're using also sometimes now in recent times also some molecules like which are proteins which are modulating the immune system. The last frontier of the vaccines are actually really the molecules similar to COVID vaccines, which are nano vaccines, so nanoparticles which have controlled size and controlled surface, and they can contain also the adjuvant inside, so you have a full control of the cargo, so-called, so the old molecule. Okay. So you can have regulated lipids which are taking part, you can have cationic lipids which are facilitating the internalization in the antigen-presenting cells, you can have helper molecules, and finally, eventually, you can either a protein antigen, or you can have, like in the case also some COVID vaccines, the messenger RNA or other type of nucleic acid. So this is really a developing world from this point of view. I talk about formulation because talking about chemically modified insulins, this is a topic which is quite important in the delivery of these molecules. Okay, as Natasha showed you before, diabetes is really a pandemic. Actually, the original projections were saying that we would reach by 2030 something like 370 millions. Actually, we almost doubled this number. We are now at 578 million people with diabetes in the world, and projections say that probably there will be an increase of up to 700 million in the year 2045, with some countries like, unfortunately, South Africa, which is increasing up to over 143%, up to 47 million in the 2045, and many other countries like Southeast Asia, China, and so on, they're going on with the same trend, also South America. Okay, and death rates, unfortunately, also very high. You see that low death rates are only in the West countries, or in the North Hemisphere countries, but death rates are very high instead in India, Africa, and South America. Okay, so the main factor for fighting against diabetes, as you know, is insulin. Insulin is composed of two main chains, chain A and chain B, okay, and can be present in the organism in different ways, as a dimer or eventually also as an hexamer. So it has a very complicated quaternary structure from this point of view. And technology has developed different type of these insulins. There are short-acting insulin, so insulin which onset is very fast after injection, and then that also decays very quickly, immediately after, in a few hours after the injection. Or you can have a middle, like the regular insulin, or the so-called isofan insulin, which are whose onset in the blood is a little bit later, 2-3 hours, and then lasts for something like 12 hours. Or you can have the so-called basal insulin, or slow-release insulin, which are like insulin detemir, which I will talk to now, or insulin glargine, okay, which is a modified insulin. These are the three main actors now in the panorama of modified insulin. This is insulin glargine, which is simply two modified amino acids in chain B and one modified amino acid in chain A. And these are the two insulins that which are chemically modified. So detemir with the tetracanoyl molecule fatty acid linked to the lysine, and the other one with the hexadecanonic acid linked to the N-terminal spacer. Okay. And what has been evident from the very beginning that the molecular size of the insulin quaternary structure, so monomer, hexamer, dihexamer, or multi-hexamer, directly affect the absorption rate of insulin in the site of injection. Okay. For instance, Sanofi Aventis resolved this problem with, as I told you before, with the glargine. The glargine has a modified 2 amino acids here, 1 amino acid here, and so it has made the molecule simply completely soluble at pH of 4. And so that's why formulation is very important. So you're formulating this type of insulin this way, you have a clear solution from injection, but you have an immediate microprecipitation in the site of the injection in the derma, which is making a kind of depot, which is causing a slow release of the hexamers, which then become dimers, and then the endomers in the bloodstream. Okay, so that's why you have this long release of glargine in the bloodstream. Okay. Instead, Novo Nordisk, who developed this chemically modified insulin, chose a different way. They modified insulin by attaching the fatty acid on the C-terminal rising group of the B chain. This is a 14-carbon fatty acid, and this kind of mechanism has a different effect because in the site of injection, sorry, here in the site of injection, the detamir is also present as a depot, but then it enters the bloodstream and binds as cargo on the albumin, and is transported in the body to the binding with the albumin. Okay, and so you can see that immediately that the half-life of the detamir in the bloodstream is the duration of the half-life is much longer from this point of view. Another way of chemical modification even improve this function. So we have the second type, which is the so-called insulin deglutide. With this type of insulin, we could reach up to 25 hours of duration in the bloodstream. And this because, again, the formulation here has a high impact on the mechanism of production. You have the formation of multi-hexamers, which are then released in monomers and enter the bloodstream. And simply because the formulation of this molecule is done by zinc, which is present in the hexamers, but also by phenol, and this is creating a kind of stacking the pot of these multi-hexamers, which are very slowly released in the bloodstream. In fact, when you inject in the site of injection, the phenol is going away, and the dimers, which are packed in the site of injection, they become a kind of polymer, okay, in the pot, which is then released to the bloodstream. Okay, so that's why I was telling you that also formulation sometimes has a very strong impact on the way a molecule is acting in the biological organism. Okay, and I will show you ICGEB project for a biosimilar detamir. Okay, as always, we are starting from both sides, from the chemical side and from the biological side. We created a system of stories in the East for express human insulin precursor. We can express around 30 grams in maybe almost maybe something more in 10 liters fermentation. Then we transform this human insulin precursor we cut the last amino acid by trypsin digestion, and then we modify with the fatty acid, which is also produced in-house. We simplify the original in bulk amounts of fatty acid and then activate with the succinylmethyl ester to react with the insulin. And we arrive at final detamir after obviously chromatography steps or preparatory chromatography step. We arrive at final yield of around 30% on the insulin precursor. So a very good product. I will show you just the type of gene which is inserted in the stories for which is then cut by trypsin to obtain the so-called BES-30 insulin. I will show you some of the in-process control data. This is the digestion of the precursor, which is very efficient, 84%. This is the attachment of the fatty acid, which is also very efficient, around 65% by HPLC in-process control. The preparatory purification, where we are collecting our molecule, our final molecule, and then some of the quality controls by which we confirm the correct attachment of the fatty acid and the insulin. So liquid control in spectrometry with the correct molecular weight, co-injection with the originator. So we buy in the pharmacy actually the originator Levemir, which is produced by Novo Nordisk, and inject in liquid chromatography mass spectrometry to confirm that they have the same retention time as mass, molecular mass. And then we also confirm that the fatty acid is in effect attached only on the B29 lysine of the molecule, such as in the originator, by a digestion and processing mapping by mass spectrometry. Okay, so these are some of the quality controls which are showing the most important critical quality attributes of the NBA from this point of view. The same thing we are doing now for Neglutec, which is the other model that I showed you before. We are actually already developed all the chemistry of the fatty acid. We have developed also all the fatty acid conjugation to human insulin and the purification. So we are particularly ready also with the biosimilar. Okay, that's all. I just maybe I'm a little bit out of time, but I will just show you again the group, okay, which I showed before. And thanks all of you for the attention. Thanks a lot.
Thank you very much, Corrado. Very comprehensive talk there.
Thank you.
Right, yeah. So do we have any questions from the audience? Zoom Master, are you able to show anything that's come across? Yeah. But so the one question that I wanted to ask was, you talked a little bit about nanotherapeutics, and I guess that's even another level of complexity, because the formulation science is also really quite it's quite something, right? So it's a specialized area. So how do you think we can go about training people around formulation science?
Formulation is really something apart, also because it's developing very quickly. So training, and also there is a lot of research running on that. Actually, in fact, many vaccines are still done in the classical way, also, as have you seen. But the type of testing that they are doing now, if you are a little bit following literature in this field, is really astonishing, especially from the point of view of the control that you can have on the construction of these nanoparticles. So you can really carefully choose the size of the nanoparticle, and by controlling the size, you can achieve a level of control also on where the nanoparticle will enter the organism. You can have control on what you're putting inside these molecules, so either nucleic acids, proteins, and so on. You can have control also from the point of view of the adjuvants, because until now we were used to adjuvants which are simply chemical moieties or whatever. Now there are kind of adjuvants which are by themselves, again, they are recombinant proteins, which is amazing, like these toll-like receptors, which are modulating the immune system. So you can pack together in a liposome, or even, they are not even sometimes liposomes, they are polymers of defined size. You can pack inside the antigen of interest together with the protein, which is activating specifically the immune system in the way that you prefer, together with the lipids, for instance, cationic lipids, which are making that enter in the cell. So it's a science which needs, I think, a very careful training, a very...
and of course, it's not just also on the manufacturer, that training, the regulators also then need quite a bit of training, isn't it?
Yeah, for sure, absolutely. Because before achieving a definitive word, before saying a definitive word on efficacy and especially on safety of some of the techniques, I think that will take years for sure, because you need very careful clinical trials before to decide which impact some of these molecules can have on the organism.
Okay. All right. So we don't seem to have questions on the one side. Just maybe one last question for you of the current vaccines. Because that's what's popular at the moment. Of the current vaccines, which ones are they? All of them nanotherapeutics?
No. The two nanotherapeutics are the Moderna and Pfizer. No, these are the two real. They are quite similar point of view of the preparation.
From a formulation
perspective. From the point of view for the formulation. There is, maybe I can eventually later, I read some papers which are quite well describing the type of molecules which are involved in the formulation of these vaccines. Maybe I can share with the audience. Maybe by
Thank
email
you.
Or whatever.
Yeah. What are the chances of us coming up with orally administered biologics?
Because
that's kind of one of the other problems.
So actually, there is, for instance, there are some for the insulins, not yet, but they are in development. And for instance, one of the main players now in diabetes and especially in obesity treatment, which is this liraglutide. Now there are oral formulations which are coming to the market. So this is a peptide which is based on the GLP-1 peptide and is modified like the insulins with fatty acid attachment. And until now, also this one was administered by parenteral injection. But now Novo Nordisk arrived to a formulation which can be given by oral administration, so it can pass the gastric system without problem and absorb to the gastric system. Also, from the pure point of view of the dosage, it seems that it can be well established, can be well equilibrated from this point of view. So it's reached the market, I think now in this month, whatever. So there is also big advancements also in this field from this point of view.
Yeah, so that I think would also increase access, probably then reduce costs.
Especially compliance from the point of view of the patient. This is the main problem. And also the fact that you don't need, for instance, you don't need a point of care, for instance, for injection and so on. You can really much more, you can distribute the pill, give clear indications on the dosage, and then you can leave the patient alone and he can do his therapy alone. Instead, always when you have to perform injections or whatever, compliance of the patient is somehow.
And I think with vaccines, what we are picking up is that there's greater part of why there's greater hesitancy is just the fact that these are injectables, because already there's a few of injections, and then for some reason people just think you'll be poisoned when there's an injection. I mean, they don't worry about what they take.
They're always suspicious from this point of view. Yes.
Yeah,
they get injected with somebody. Yes.
Also, if I can add, Corrado mentioned this oral insulin. So New York-based company Oramed, they launched these huge clinical trials for oral diabetes. So this is something that is going on. So they launched the largest and more advanced clinical trials under the FDA, and they involved the huge number of patients. So we are there. It's almost there.
Oh, that's great. Yes. So what we're now going to do is we're going to do like a 5-minute, discussion Q&A. That's if there are any questions from colleagues here or on the call.
If I can, David, if I can, I wanted just to come back to your question before United States. I remember, because we discussed here a lot about that the primary, why still they do not have all these approved biosimilars in the US market. And one of the big reason was the litigation between companies. So remember, Corrado, that you were saying like the primary reason for this delay market was ongoing this patent litigation. So I remember that we read a famous case of Sandoz, Milan, Biocon. They finally reached this global settlement with these companies, AbbVie and Genentech, because regarding claims that the biosimilars adalimumab, and it was also trastuzumab, infringe some patents that protected from originators. So even if it's FDA approved, they still can have years of litigation with the company who was originator and trying to, of course, protect its market.
Yeah, okay, so that brings up the issue of IP. Because I think there's more contestation between companies around IP than we actually realize. So how does ICGEB deal with the IP transfer issues? So if somebody came to you and they said, we would like to take transfer,
As we are not producers, we do not produce. So we have a technology transfer office who can help us in some IP issues when it comes to some new products that ICGEB is developing. But we really, when we offer training, we always said we do not, like, the due diligence has to be done by the company, because it's a huge field. It's like every single molecule is protected, like more than 200 patents. So it's not just saying patent expired this year. They have, like, even now when all these famous cases came out, like adalimumab, of course, because it's one of the main drug on the market, and the market is huge. They came out that the originator put also some patents afterwards to protect the formulation, to protect this and some. So it's really complicated field. So we never enter in that field. We transfer knowledge. We always say we really help to acquire the whole, as Corrado mentioned, it's really to be innovative, to put one production process. And that's the reason why we never enter in the formulation part. So we always finish training and development with a concentrated protein solution. Then this is when we finish all the quality control tests with the reference with the standards. Then every company, it has to work on its own formulation.
Okay, so in other words, your tech transfer is really about the API. Let's call it that.
Yes.
So it's really about how do you get to the point of the API. Then thereafter, that company would then have to navigate its own way around the formulation.
Of course, we formulate it in order to keep that, and there's some standard formulation, and then we compare it to the reference product. But in order to make really biosimilarity assessment, the company has to decide what the formulation of the drug will be, and then repeat all the tests that we teach them to with the reference standard on their own market.
Okay. No, so that's good then. Do you want maybe give me an idea of what sort of uptake you've had in terms of technology transfer since the program started? What are the sort of successes that you've had?
So our success is any kind of training. So it doesn't depend on if the final product is actually on the market, because we are not following our guests until the end, because we just cover, as I show on that slide, we just covered the first part. So we are developing API, and then they can optimize the process, they can scale it up, they can do whatever they want to change, improve it or whatever. But for us, the main mission is like the transfer of know-how. And this is what we really did very well. So we did, even now during the COVID, we had 3 guests that we signed 3 contracts with companies from 3 different countries, where they really want to have their people trained, and they have a basic idea, and then they can. So for us, success means we transfer knowledge. So how that will reach the market, it really depends on the company. So their own business plan, their capacity, money, or whatever, or regulation part in their own country. We have 66 countries that we work with. So it's like member states. So it's really hard to follow all these regulations and help them in that part. So we live on the first part.
Mm-hmm. Okay, and then in terms of sort of therapeutic areas, which therapeutic areas are you finding that your partners are interested in?
So it really changed over the years. So as Corrado was showing you, like for the beginning, we started with like 25 years ago, we started with interferons. Then interferon stopped to be of the interest because other drugs, small molecules, came out for the treatment of hepatitis. Then GCSF was one of our molecules, and it was really still molecule that people have interest, although there are many biosimilars already registered on market. that's the reason why we went further to chemically modify them, because then it gives the advantage, like having, as Corrado mentioned, administration once a day or once a week or something, it's really get more advantage. And lately, what we can see that people are even more and more interested in insulins. So, and now with the new facility, we really would like to move more towards the monoclonal antibodies or the fragments. So we are now working on the two molecules. One molecule is trastuzumab in the mammalian cell, production in mammalian CHO cells, and the other one is stertrolizumab pegol. It's a pegylated molecule produced in E. coli. And a part of it, we were really working and gained some experience on the molecule that Corrado mentioned for diabetes and insulin. It's a molecule, liraglutide, that we are now trying to develop the chemical synthesis of this molecule.
Okay, well, that's great. Then, so you know what I like about biosimilars is the fact that you're not moving anything, so you're not going to be importing API, because that's the problem we have in the sort of small molecule space. So, for instance, in Africa, we don't have the capabilities of synthesizing APIs. So we are constantly having to bring in API from actually competitor companies. And these competitor companies can bring in their products tariff-free, because finished pharmaceutical product can come into a lot of African markets with very little tariffs on them. Whereas with biologics, you can say once you have the cell culture and the cells and you're looking after them, you're actually making the API in your own countries. But now the question I need to ask is, in terms of those cells, do you provide these as part of the tech transfer, or you are then pointing whoever is wanting to, your partner or your client, to the sources of these cells, the starter cell cultures?
So I have to say that these cells are made in our lab, that are R&D labs. So that was the main issue, because the cells has to be good manufacturing in GMP conditions. They have to be made in the GMP. And when you are doing with E. coli and the yeast, we teach people how we make that cell, so they can make their own cells and they can decide where to take it and how to proceed. And of course, that with mammalian cell, now that we are moving to the monoclonal antibodies, this is really complicated issue, because you have to spend at least 6 months to 1 year to obtain a good producer cell. That is 1% of all the cells that you have you genetically modify, and that's the reason why we now invested in the robo. We have a clone pick. We have a machine who help us to pick the clones, and it can do 10,000 clones in a week, and we can then test much more than before, like with the serial dilution and so on. So now we are aware that if we are moving to the mammalian cell, that the issue starts to be more complicated, because you can't tell people, oh, you make it on your own. So now we are trying to see how to go in this pharma compliant field, and how to make the cells more GMP, and then have different.
Okay, so in that case, that would then become another revenue stream for you. In other words, I could then come to you and say, "Can I get Chinese hamster ovary cells from you?" And then you would, sell that to me. And then I would from there multiply them.
You have to make a GMP, you have to do GMP characterization of your master cell bank and so on. This is really a good point. This is a very important point. I also prepared, if you allow me, if there is no more questions, like the conclusion just of all these both talks, and just to have a take-home message, I have a few slides to show.
Yes, that would be great. I don't know if there's any last words from Corrado on his presentation.
No, I mean, it's want just to say that I mean, the chemical modification is something we really adds value to the biosimilar market from this point of view. So it's something we should be taken in consideration even from small companies, because the chemistry involved maybe is complicated, but keep being afforded in, especially chemical synthesis can be afforded in large scale, which quite small investments from this point of view, okay? So to produce grams and grams of monoclonal antibodies can be demanding from this point of view, but to produce hundreds of grams of a chemical modifier is not. Okay, so it's something we should be taken in consideration when approaching to the projecting of which biosimilar to choose, for instance. Okay, so it's something we should take.
But these things are so potent. I mean, like if I look at it's milligrams, you know. So if you look at something like Actemra, what is it? I think it's in a 400 milligrams in 20 mils. So it's extremely potent, and you're paying $500 for that. So I mean, if you were to produce 2 grams of it, you could probably retire on that.
Yeah, sometimes it is the added value of the chemical modification is a lot, actually. Yes, you can see, for instance, for filgrastim, if you produce filgrastim, it costs a few dollars a dose. You produce filgrastim pegylated, it immediately goes up of 2 orders of magnitude in price on the markets. So the price on the chemistry that you're using is absolutely not comparable to the increase of money that those fund. So it's something we should be really taking into account if you're projecting to invest in biosimilar markets.
Okay, thank you very much. So we will close off then with Natasha summarizing everything, and then Mampe will come in with final remarks. So Natasha, back over to you.
Okay, thank you very much, David. So just this slide to remind you, as David said, like, yes, if we talk about local production importance, all African countries are net importers of medical and pharmaceutical products. It's estimated that continent cover 94% of its pharmaceutical needs through import.
Natasha, could you put this up as a full screen?
Yes, sorry. Yeah. So, and then many of the countries provide these pharmaceuticals are heavily disrupted by COVID. We saw COVID-19, and moreover, at least 94 countries in the world have now also restricted exports of medical supplies as part of their response to COVID-19. That these were all data from last year, but this is something that we should really keep in mind, and when we think if we ever question if local production is important. So I wanted just to say a few words what we heard today. So we heard today about biologics and biosimilars that are produced using different expression system in the complex manufacturing process, that even small changes or differences in manufacturing processes may have a significant impact on quality, purity, activity of the final product. Comparison of similarity between the biosimilar originals, as we heard from Dr. Aguinancia's presentation, continue to be a challenge. Biosimilars are set to play a key part in the future healthcare. So understanding this opportunity is key for pharmaceutical companies competing in the emerging markets. And biosimilars potential is, as we heard and we saw many examples, is to reduce price and expenditures and increase availability. And there is a robust regulatory framework for biopharmaceutical drugs, but the harmonization promotes collaboration in more rapid access to medicine. As we said, we think that sharing knowledge, sharing what we learned can help our member states. Technology transfer can accelerate acquisition of this knowledge, experience, and equipment necessary to build a strong local pharma sector. ICGEB is actively involved in strengthening these capacities and competencies of our local partners and transfer of suitable technology. So we share knowledge, we transfer know-how, and we train people online or in-house for production and purification of those and biologics. And I would like to thank everyone for their attention. I hope it was useful, and then we succeed to transfer all these messages. And I would like to invite you for tomorrow's part of this workshop that will be presented by Dr. Michela Tertoslavic from my group here at ICGEB, and she will present designing a manufacturing facility for biologics biosimilars. She will talk about good manufacturing practice requirements and quality assurance principles that can be applied to any biologic, vaccine, and biosimilar. Thank you very much for your attention.
Yeah, thanks, Natasha. So over to Mampe. Yeah, but don't forget to join us tomorrow as well at 12 to 2 o'clock. Over to you, Mampe.
Thank you very much, Dr. Natasha Skoko, Dr. Aguinancia, Dr. Katerere, for a really insightful presentation. We have 39 participants currently online, and I hope they are from all corners of the continent and that they have had the really brilliant presentation that you have made today. I think your last few slides really summarizes what we're trying to achieve as the DSI working in the continent. We do realize that we need partnerships, we need support, we need to be capacitated from a technical perspective as a continent. And from what you said, Dr. Skoko, the ICGEB is always ready and willing through its members in the continent. I think we've got about 16 affiliated ICGEB members in the continent. And it would be a shame if those 16 governments do not take advantage of the opportunities that the ICGEB has to offer. So we try to bring this kind of training into this platform, but there's nothing that stops each of the 16 ICGEB member states to have national technical workshop on these topics or any other topics that the ICGEB can offer. We are really trying to address the health challenges that the continent faces through the production of medicines or vaccines from the continent. And the whole theme of this conference is aimed at doing just that. So as you said, Dr. Skoko, the meeting tomorrow, the training session tomorrow will be very useful also for participants, and we again ask them to join in. We also have a session that starts between 11 that speaks to how we can support the African Continental Free Trade Area in relation to Africa producing its own medicines. So we urge participants to also join in at 11. It's track 6 tomorrow. And of course, a follow-up session on the training will follow at 12:30, if I'm not mistaken, until 2 o'clock. And as indicated, the focus for tomorrow is on GMP requirements, General Manufacturing Practice requirements. So please kindly connect and join in, and we appreciate your joining in and staying till this late. Thank you very much to everybody. Take care.
Okay, so we'll see you tomorrow. The session for tomorrow will be particularly interesting and important for regulators as well, because there's a GMP component, especially GMP inspectors and colleagues in that area. So make sure that you invite other people. And I think this is also being recorded for future access. So with that, I'd like to wish you well and be safe. So I'll see you tomorrow.
Okay.
Thank you.
Thank you. Bye. Bye.
Thank you. Bye-bye. Thank you.
Yeah.